Introduction. It's a pleasure to be here to present the work of our lab on reconstructing the regulatory network of TB and its hypoxic response. So just to bring everyone onto the same page, tuberculosis, which is caused by Mycobacteria tuberculosis with a genome size around 4,000 genes, kills between 2 to 3 million people per year, and it's mostly happening in the developing world, but it's also the primary cause of death in AIDS patients. More importantly, despite the fact that we didn't have a single drug for TB just 50 years ago, we now see this recent emergence of drug-resistant strains. For example, the XDR, or extreme drug-resistant strains, are basically resistant to every drug that is out there. And these cases have been reported all around the world. Now, part of the reason that TB is such a successful pathogen has to do with its pathogenesis mechanism. So as you all know, TB is inhaled. The primary route for catching TB is through inhalation. It's inhaled through air, and then it's uptaken by macrophages. And in most cases, the immune system is able to clear it up, but for some cases, TB is actually able to persist inside the macrophages and go on into this latent form, latent form, or which is an asymptomatic type of TB, and remain in that state for several years. Later on, when the immune system is weakened, for example, by AIDS or aging or malnutrition, it can actually be reactivated. Now, the scary statistic is that 1/3 of the world is known to have latent TB. And this is why TB is still quite a significant problem, and TB latency is now the focus of much of the ongoing research on TB. It's not really completely understood. It's known that it involves some metabolic alterations, such as adaptations to hypoxia inside the granuloma and shifts to lipids, including cholesterol. And very little is known about the regulatory programs that drive these adaptations to hypoxic conditions. DOSR is a major regulator known to induce a few hundreds of genes, but the full circuitry is not really well known. So this brings us to our systems approach to try to piece together the circuitry that underlie TB pathogenesis, and in particular its latency. And ideally, that will mean that we would like to develop predictive models, not just for the metabolic network, but also for the regulatory network of TB. This is done through a collaborative effort between Boston University, Harvard MIT Broad Institute, Stanford, Seattle Biomed, and several other labs to do comprehensive profiling on transcriptomics, glycomics, several omics data, and ChipSeek of TB regulators, not just on in vitro cultures, but also from macrophage cultures. Our lab in particular performs the ChipSeek of TB regulators and performs all the computational analyses and integrations of data sets. So the first part of this talk will focus on ChipSeek as a method for identifying regulatory bindings in TB. Just a quick overview, chromatin immunoprecipitation followed by sequencing is a well-known method for identifying DNA binding proteins and the regions. So the protein of interest is cross-linked to DNA, and then a specific antibody to that transcription factor is used to enrich for protein-bound DNA fragments. The fragments are then aligned to the genome reference, and giving a distribution or a coverage in the regions where we have binding of the protein to the DNA. So this is a typical example of our TB chip binding event. It shows the forward in red, the green, the reverse in green, and the total coverage in an intergenic region for the chip performed for the TF KSDR. KSDR is known to be important in the TB lipid metabolism. And this figure below shows the genome-wide chip of the same transcription factor. We see a typical background coverage of around 150-fold, while we do see peaks as high as 1 million-fold coverage. Our ChipSeek system is performed using an inducible promoter system designed by Kyle Minch and our collaborators in Seattle Biomed. This system involves flag tag transcription factors induced by a tetracycline promoter. And using this system, we avoid We avoid using specific antibodies for each of the transcription factors, and thus this allows genome-wide or large-scale ChipSeek for any organism. And up to now, we have performed ChIP for 51 regulators out of around 180 TB regulators, which is about a quarter. This data set has been analyzed using a pipeline designed by Matt Peterson from our lab to identify the significant binding sites and regions from the coverage, ChipSeek coverage data. This figure here shows the peaks ordered decreasingly for KSDR. The red bars show the previously known binding sites for KSDR, while all the blue bars show the newly identified sites. And as you can see, the majority of the novel sites have actually higher affinity than the weakest known binding site. And this extensive low affinity binding has indeed been observed in several other organisms. Now, the questions that we would like to ask about this binding information is, first, are these bindings real or just random globbing to the DNA? Are these binding events happening at physiological levels of the transcription factors? Would we see the same bindings in different conditions? And finally, do the bindings have regulatory functions, and are they predictive of gene regulation and expression? So starting from the first question, are these bindings real? Anna and Antonio from our lab have shown that binding is reproducible, meaning that in this figure, for example, you can see the distribution of the distance between corresponding binding sites into ChIP replicates of KSDR, as well as two other transcription factors. You can see that most of the distances are within the length of the predicted motif. And more than that, we see that the peak heights themselves are also correlated between the replicates for these transcription factors. Also, binding is shown to be associated with motif strength. For KSDR, for example, we see that all the strong peaks are associated with the canonical KSDR motif identified before, while for the weaker peaks, we see degraded versions of that motif that lack accessory bases known to be important for shaping the affinity. So these results together show the validity of our binding information, and for more details about these interesting analyses, you can actually check out Anna and Antonio's posters on Tuesday. The second question is whether these bindings are happening at physiological levels. For addressing this question, we did comparisons for a few of our transcription factors. For the case of DOSR, which, as I mentioned, is important for TB hypoxic response, we compared the peak heights for different induction levels of this transcription factor. And we saw that for the highest induction level, we see a lot of weaker peaks revealing, which we couldn't be able to see with lower induction levels. We also compared the actual expression level of DOSR in each of these induction levels to its expression level in hypoxia as its physiological stimuli. And you can see that the levels are very much comparable. But more than that, for the DOSR regulon genes themselves, the expressions of the genes with our system, shown in red squares, are very much comparable to their expression values in hypoxia, using both our system in green and with the native promoter shown in blue. So this together shows that, at least for DOSR, all these additional new peaks are physiological. As in another example, RV0081, another transcription factor, which actually has the most binding based on our ChipSeek data, we compared the levels again to its level in hypoxia. We can see that it's very much comparable. Actually, 80% of the binding sites of 0081 are detectable at the physiological levels. We'll talk about this transcription factor later on more. We have also further validation from other literature. So, for example, the Blasco et al. Paper, which has performed wild-type native antibody chip for regulator ESBR, shows, comparing it with our results using our inducible promoter system, shows that not only the binding sites are very similar, but also the relative strengths of the bindings, the motif that is identified, and also for both cases we see abundant genic bindings of this transcription factor. Finally, the ChipSeek data shows us where the binding locations are, and it gives us information about the binding sites. But we don't to know whether these bindings have upregulating or downregulating roles. We have combined this with comprehensive transcriptomics data. So this TF induction data was done, performed alongside with the ChipSeek, such that each of the experiments, microarray experiments, were the induction or overexpression of a transcription factor, and we have measurements for all 4,000 TB genes. Surprisingly, we see that RV0081 drives the most changes in the genome. And this is very much consistent with what we saw about the highest number of bindings for this TF. So one way to assign regulatory roles to a TF gene interaction is to compare the expression of a gene when that TF is induced to a background distribution of the gene expression. And as expected, we saw less regulatory information for weaker peaks, and while the stronger peaks and intergenic peaks had more regulatory roles. We assign potential regulation to 25% of the entire gene peaks, and we do not identify regulatory roles for all of the previously known binding sites, which shows that our method is still conservative. But as you all know, some transcription factors work together in combinatorial activities to regulate genes. Therefore, we took a step further to do modeling and relate the expression of genes to the expression of their transcription factors that bind to that gene based on ChipSeek data. We trained these models on independent ChipSeek and normoxic TF induction expression and tried to generalize these models to test the predictive power of these models in an independent data set of 7 days of hypoxia and 7 days of reaeration time course. For more details about how this modeling was done, I have a poster you can check out downstairs, but as a summary, we could predict the time course expressions of a diverse set of genes. From central metabolism, lipid metabolism, as well as major transcription factors. For example, KSDR, we could predict its induction and activation in hypoxia. So we have shown that our binding information is real, functional, and predictive. Now the next step would be to relate it to the biological problem of TB latency. So in a global scale, this is how the TB regulatory network looked like around 2 years ago. With our a quarter of the regulators being chipped, we now have this huge hairball, revealing thousands of new interactions connecting 2,700 genes. And we also see a lot of interactions between the regulators themselves, shown in red circles, so that some of the transcription factors basically mediate interactions between the regulons. This figure shows a subnetwork of some of the major regulators in hypoxia and redox adaptation, the enduring hypoxic response, chromatin modeling, and cholesterol and fatty acid degradation. And our network actually links these two factors, hypoxic regulators and the enduring hypoxic regulators, to KSDR, which is the cholesterol and fatty acid or lipid metabolism regulator. So these two factors that were thought to be involved in TB latency independently are now linked through this network. To test this, we did further profiling. we performed metabolomics and lipidomics of the same time course of hypoxia and reaeration to test whether hypoxia actually affects lipid and related metabolism. This figure shows an overview of the MTB metabolic network. The changes in gene expression and metabolite changes are shown in heat maps. I'm not going to go through the details, but in summary, we see activation of cholesterol degradation genes. We see a buildup of triacylglycerates, an induction of the methyl citrate cycle, buildup of extracellular amino acids as potential carbon source. And the fact is that there is an underlying program of all these changes in the genes from the hypoxic regulators as well as pH and redox regulators. These include 0081, KSDR, YB3, DOSR, and so on. Our lipidomics data also confirms the significant changes in lipids as well as glycolic acids. And these are actually important immunomodulatory factors in the mycobacterial cell wall. So in summary, adaptations to hypoxia, propionate, and redox stress inside the macrophage, as well as the consumption of hosts and internal lipids or potentially amino acids, lead to the production of immunomodulatory lipids. And from there, again, we have feedback to production of foamy macrophages. And again, this all gives feedback to TB persistence and its latency. But of course, these are just initial interpretations based on a quarter of the MTB regulators, and we are currently extending that to the entire 180 regulators. But the systems-wide nature of this data in general gives the opportunity for further investigation and more targeted experiments. Given that, we have released all of our data for public access on TBDB. Finally, I'd like to acknowledge ISCB for their generous travel award that enabled me to come here. Our collaborators, the Sherman Lab, Skolnick Lab, and several labs that produce and develop the data that we're using, and my advisor, James Galligan, and all the members of the Galligan Lab. And thank you for listening. Any questions?
So when you look at the integration of ChipSeek data and gene expression data, is it possible to collapse the gene expression data per regulome, I mean, combining multiple genes?
So the modeling that we performed and the assigning of regulatory roles to the bindings was with the same aim to combine the ChipSeek and the regulation. But we did actually do further analysis of clustering of the expression time course expressions to see how the regulators are affecting the hierarchical clustering or divergence of expression using a DREM algorithm. And we did identify that some of the regulators, such as 0081 and DOSR, which are hypoxic regulators, are involved in the branching of the time course expressions. That's what you're asking about.
So I have a question. It's just the closest microphone. I was wondering, the results looked really nice, and I liked the predictive model that you developed and then validated. I was wondering if you could maybe just go into that briefly about what technique you're using to do the predictions and the transcriptomic responses.
So it involves a statistical model selection to, first of all, find the best model structure that relates the expression of a gene to the expression of the transcription factors. They are all linear regression models, but some of them have, for example, interaction terms and other additional or optional terms. And the best model structure was selected, and also a subset of the binding regulators, of the binding transcription factors were selected as regressors or regulators. And again, that was done through an iterative process of model selection and training.
So I also have a question.
Yes.
I was wondering if some of your model building and validation is focused on interrupting not just the infectious disease part, but also the latency, where the bacterium is sort of dormant within the host, but not necessarily infectious.
Right. If you're talking about the macrophage cultures,
Right.
those analyses are something that we're working on right now. We currently have RT-PCR data, time course data for cultures inside macrophages. And we would like to, again, compare them with the results that we have from in vitro TB.
Mm-hmm.
Well, it's more of a philosophical question maybe at this point, but you have shown a huge network, right?
Right.
Several thousand genes, that have been implied as taking part in this process. But how many of them do you think? Well, what's your take on it? How many of them are actually required for the hypoxic response, and how many of them are just a side effect? So you see it goes up or down, but it doesn't have to. It just does it, and if it didn't, well, the response will be almost the same, right?
Yeah.
So is it like, out of the 3,000 genes, will it be, 20, 30, 1,000? I don't know. What's your take on it? That's the question, I guess.
So first, the focus is on finding the regulators that are kind of driving this response, hypoxic response, or the changes. And we have at most 180 TB regulators. But the way that we look at it in a systems view is not just a specific transcription factor making a change. It's a combination or a circuitry of regulatory programs that together they make or drive regulatory programs or changes in expression that lead to TB latency, basically, if I've answered your question.
To comment on that, so you actually have two sort of problems here. One is the specific context. So, of course, when you do a ChipSeek, so you want to gather as many regulations that you can. But if you really have very fine time course, you would see, okay, only 10% or 5% of genes are upregulated as direct regulation, and the rest are probably more downstream sequences.
Indirect regulation.
Yeah. The second point is that actually it's quite common that under this kind of from pneumoxia to hypoxia is massive reprogramming of the cell transcriptome. So it's very common to see thousands of genes are changed. That's very normal.
Thank you for the comment.
Great. Well, thanks, Elham, for a nice presentation.